Good morning, everybody. Thank you very much for the organizing committee. It's a very great honor for me. Invite me as one of plenary speaker in this first international conference on biosciences and biorefinery 2020. And actually, this is really great time, even though that I did not do a lot of work here, but let me in this occasion time, I would like to spread the idea about how the bioinformatics can accelerate the biomedical research. Here, I propose the one of applications of bioinformatics is the use of the reverse vaccinology. So let's start with what we say about the classical vaccinology is that in the beginning, at the time that the scientists is use the microorganisms itself, they try to grow the pathogens, and then they try to modify using the attenuated vaccine or the inactivated vaccine based on the microorganism, and then they try to inject the microorganisms to induce the immune response systems for the human. And this is what we call that the term of the classical vaccinology. And then when we go through the time that starting from 1930, that as the first foundations of the classical vaccinology, that many diseases try to cure by using the empirical approach, such as diphtheria, tetanus, pertussis, rabies, influenza, smallpox, polio, BCG. And then around 50 years afterward, that in 1980, the scientists start to work with the recombinant DNA. It starts from hepatitis B, cellular pertussis, the Lyme, and also the human papillomavirus. And then 10 years after, the vaccine technology improved, that it started to use the glycoconjugations in 1990. It starts from meningitis A, C, W, and Y, also for the pneumo, for the HIV, GBS, and also for Staphylococcus aureus. And around 20 years afterward, the scientists also start to improve the technology using the reverse vaccinology in 2010. It started with the development of vaccine for meningitis B, the GBS, GIS, for E. coli, for Staphylococcus aureus, and also another bacteria. And again, like by pandemic COVID-19 conditions, is also we have a breakthrough also in the vaccine technology, such as the synthetic vaccine using the mRNA technology. But today I would like to focusing on the reverse vaccinology as my topic. So the idea from the reverse vaccinology is try to design from the in silico. We try to analyze the sequences, the genome informations, the proteome informations, or the any genetic information that could be utilized by us. And then we can try to design the vaccine candidates, or the PVC, the potential vaccine candidate, and then we can express it to the recombinant proteins. And what will be this is the breakthrough of the reverse vaccinology approach, that in 2015 for the meningitis B, the first vaccine is to receive therapy designation by FDA. And as you can see here in the Princeton, USA, they start to begin the meningitis vaccinations. And also in the London, UK, that since September 1st, 2015, all newborns in UK are vaccinated with meningitis B vaccine, which is produced by using the reverse vaccinology approach. And as you can see here, that this is the report from the clinical trial in Canada in Seguin-Lac-Saint-Jean in 2014. They tried to vaccinate the entire population from 2 months to 20 years of age. And as you can see, around 43,000 people were vaccinated between May to June 2014, and around 12,000 are followed with active surveillance. And as you can see, around 14 to 15% of has a fever in less than 2 years. Less than expected, but high use of antipyretics, 2 convulsions, which are 6, are expected, and 99% willing to receive the second dose. And the active surveillance showed that they not identified any severe problem and confirmed significant frequency of local pain, fever less than expected, and general malaise. And as you can see, that no invasive meningitis B cases observed among the 47,000 people vaccinated from age 2 months to 20 years, with the crude of vaccine effectiveness is about 100%. So what I showed to you, this is the quite promising, although that this after this discovery of the meningitis B vaccine, there is not yet another vaccine that produced using the reverse vaccinology approach. This is the real efficacy from the field, that the efficacy from the field showed that around 83% effectiveness and 94% efficacy during the first 10 months in the UK. So this is show that even that here in the UK, for the meningitis B vaccination, it is supported by many audience, even though this is still we can say that the first vaccinology is still the new technology, but since they see the real efficacy from the field, and so many people are willing to take or to get vaccinated here. And yes, this is that this is the an example that we can eliminate the meningococcal meningitis. And of course, we can try to extend the research also to provide the vaccine even for antibiotic resistance bacteria or superbugs, for Staphylococcus, E. coli, Pseudomonas, and other bacteria. So this is also that. Then here, even with the COVID-19 pandemic, previously we have also some disease problems such as malaria, but fortunately malaria on 6th of October 2020 from the WHO, it's recommended for widespread use for RTS as malaria vaccines. The first malaria vaccine license with the AA01 adjuvants actually already reported on 2015, but then 6 years later, I guess around 8 days, from on 6th October, the WHO is recommended for widespread use. This is quite very good happy. So then the vaccines is continue to serve our society, and so what is will be the next generations or next generations of vaccinology is we try to move from the very classical vaccinology to the reverse vaccinology. Of course, the abundance of database that come from genome informations, also from the proteomic, from the structural, also from the antibody variations, and also from the immune response that can be mapped and we can analyze together, so we can improve the vaccine, the development of vaccine technology. And what actually the methodology behind the reverse vaccinology is that, of course, we need the informations. The information that can come from the genomics or from the whole genome sequencing approach, and then from the proteomics, proteome of the virome, for example, and also from the antibodies itself or from the structure. So, this is what we call the top-down approach. And then we will use the computer-based approach, so we can find the candidate vaccine antigen here, which is that from the potential vaccine antigen itself, it can be highly sensitive. And of course, this is not hypothesis driven. So we don't know which part of the genome that could be have the antigenic response, the immunogenic response, the allergenic response. But we try to analyze all the data. And we try, after we got the result, then we start to think about the hypothesis. And in this case, we no need to grow the pathogens, and then we can identify the antigens as independently. And as for bioinformatics protocol itself, that what we need to do that in order to use the reverse vaccinology is that we need first to predict all the open reading frame itself. And as in a bioinformatics approach, we can use it based on the homology, or we can try to predict using the ab initio prediction, so using the de novo predictions. And then we can try to combine also for the ORF prediction with the other computational base, such as the data mining approach. Here I mentioned about we can do the filtering approach. Then we can try to filter the data using the decision tree approach. This is the algorithm also that applied to make the lineage, the Pango lineage of SARS-CoV-2. That's why previously we know that for the UK variants, it's called the B117, and also for the Delta is B1617.2. This is come from the filtering process on the computational based approach, the decision tree. And also what we can use is also the classifying, which is we can use the machine learning. Then we can get the potential vaccine candidates, such as we can get it from the membrane proteins, we can get from envelope, the secretions, or even from the repair protein. And then Next, we can start to analyze about the virulence factors and also the essential genes that could be useful for the next experiment. So then, what is the challenges of the reverse vaccinology approach? Of course, that since we are depending on the conservations of the sequence, so information itself is very, very important. So what we need to keep in mind that there are the structural and variations in individual antigens. And also we need to aware about the population biology and the evolutionary studies such as that already mentioned by Professor Matsuda regarding about how the in the future that variants can be more and more come to the human populations, and also the population approach to the vaccine assessments, and also the need of the genomic disease surveillance. This here we need to understanding and combating the violence and also the vaccine escape, and the genome sequencing vaccine design, and also for the assessment. So let's start with one of the case of study that we have many times that we understand that the pandemics it's always become a threat, not only for today but also for the future. And when we understand, when we can cope for the disease itself, as you can see that there will be a big problem, especially for the poor income countries. As you can see here, the poorer the country, there will be the life expectancy also will be lower comparing with the GDP per capita. And as you can see also in the graph in the downside, that for more higher the country income, as you can see, the problem for the higher income countries is the non-communicable disease. But for the lower income country, the problem will come still from the communicable disease or from the infectious disease itself. So this is something that also in Indonesia still will be the challenge. And as you can see that from the Indonesia, the top 10 causes of death is from 10 from cardiovascular, and also we see from the infectious disease come from the respiratory, also for the chronic respiratory, and also the enteric infections. And we need also to understand that vaccines can prevent an estimated is 2.5 million deaths among children under 5 years, and still one child dies every 20 seconds from disease that could be have prevented by a vaccine. So what we have done using the vaccine reverse vaccinology approach is we develop the in silico prediction here. And as you can see here, I try to make it brief that here we can start from the sampling and sample selections, and then we can do the selections of B cell and T cell epitope candidates. We can apply several parameters such as antigenicity, allergenicity, toxicity, conservation, and other parameters, and then we can do the docking to the HLA to the human leukocyte antigen from class 1 and class 2. And then we start to make the vaccine constructions. We can do the single or multi-epitope vaccine with the additional linker and also adjuvant here. And here we did the vaccine docking with the receptor. And in the last part, that what we did here is that we did the in silico cloning, so we can get the plasmid reconstructions in the vector expression system. So this is something that we can optimize using the bioinformatics approach. So this is the case what we have done with using in silico prediction using the HIV1. We try to retrieve the sequence from Indonesia, and then we try to map. Then we get around 700 sequences here. And then we got it from the envelope and also from the GAG, P17 and also P24. And then we try to map it to the MHC class 1 and class 2 from Indonesia. And we got the origin of MHCs from the Sundanese, Yogyakarta, Javanese, Sundanese, Maluku, and also the entity from Nusa Tenggara Timur. And from the sequence entropy analysis, we try to check, as you can see, that the red color show that the entropy is quite low for some regions in the P17 and P24. And then we try also with another proteins, GP120. As you can see, the comparisons between the P17 and GP120. In GP120, we cannot move too much to predict the epitope because the entropy due to the mutations at that regions is quite high. So here we try to make the filtering here, the epitope with conservations more than 85%. We got the total at the end is 135. And when we applied for the MHC class 1 analysis, we filtered some of epitopes, and we finally reduced to the sum of 13 epitope. And we applied also for MHC class 2 analysis. As you can see that from the 13th, we go down to the 10 of epitopes candidate here. And then we try to check with the self-peptide analysis. We go down to the 9 of epitopes here. And then we use the IEDB, International Epitope Database. And finally, we come up with the 5 epitopes.
Sorry, your time is only 1 more minute.
Okay, I will finish. Okay, so after that, we try to check the antigenic activity, and we make a comparison using the human negative serum and also the blank solutions, and we see that the preliminary study is, we can see the absorption value is constant, even though we have different epitope concentrations. And here also we have another preliminary data that the absorption value is decreased, follow the epitope concentration here. So for me, still, of course, this is if we compare with the work from the Matsuda Sensei or even Ibu Erna, still a long way to go for future direction, especially to prove the concept of or the result of predictions for the epitopes using the reverse vaccinology. So this is something they need to go in the future. So this is the acknowledgements for all the teams that contributed in this research. So thank you very much for Bu Marcel as a moderator, and thank you for the organizing committee. Thank you.